Hello, I'm Monica Donegan. I'm a PhD candidate at the Almeida Lab at UC Berkeley.
And I'm Andrea Brown, also a PhD candidate in the Almeida Lab.
Great, and this is a video for the CDFA final report for a project we did in Hopland, California. So the causal agent of Pierce's disease is Xylella fastidiosa, specifically subspecies fastidiosa. There's lots of strains within California, and we've shown in the past that they are genetically distinct from each other. And so in this project, we wanted to test two genetically distinct strains of Xylella fastidiosa. And for the project, we picked one from Mendocino County, specifically Hopland, California, which is a town within Mendocino, and another from Bakersfield. And from the phylogeny, you can see that these two strains are distinct from each other. We had a research site in Hopland, California at the Hopland Research and Extension Center. And from the map on the right, you can see that there were 13 different cultivars of grapevines. They are all the different colors. These are cultivars that came from the Mediterranean region, and they were originally planted to test how they did in California. And we decided to infect them with Xylella to see how Pierce's disease developed in this region. So in May of 2021, we inoculated over 480 vines across these 13 Mediterranean cultivars with two distinct strains of Xylella fastidiosa. One strain was the local strain from Hopland, California, and the other strain was the non-local strain. And here's some pictures of my colleagues, Isabel and Rodrigo, in infecting vines. And then you can also see the B and C show different cultivars. Some of the different cultivars were at different growth stages when we did the inoculations, just because they have different timing and phenology. So what we saw across multiple winters, so we infected all of the vines in 2021, and both the Hopland and the Bakersfield strain initially infected vines at similar rates. But after the first winter, so in 2021, so in A on the left, you see that the Hopland strain survived the winter at greater rates than the Bakersfield strain. And again, after the second winter, so in 2023, of vines that were positive in 2022, again, the Hopland strain was surviving the winter at greater rates than the Bakersfield strain. This was interesting because there hasn't been any differences between distinct strains of Xylella shown for overwinter curing before. And another interesting part of this was that there was actually more, there was lower survival in the second winter than the first winter. This is surprising because usually we would think that the bacterium has more time to replicate and to move across the vine, but there was actually lower survival in the second winter. This may be because, as you see in B, B depicts the hours under 45 degrees Celsius that accumulated across the winter. And so the purple line was the second winter from 2022 to 2023, and it was a colder winter than the first winter. So winter temperatures may play an important role. The bacterial population, so this is how much bacteria we could detect in the vine, also changed over time. And in later years, we detected lower populations, especially in September, which makes sense as they're being knocked back over the winter. So the other factors that were important for overwinter recovery were the bacterial population size in the previous year, the Pierce's disease strain, so the Hopland versus the Bakersfield strain, and then also the variety of the grapevine and the year. So this is showing recovery in vines across 2022 and 2023. So in vines that recovered, so the kind of teal color, they had lower population sizes in the previous fall. So you can see the ones that survived the winter, the red bar kind of had greater population sizes. And this trend held true across both winters. So the population size in the previous fall is important for whether or not the strain is surviving the winter. And then also these on the x-axis, we have all 13 grapevine cultivars that were in the study and the recovery in 2020 in blue, and then in 2023 in gray. And you can see that there's big differences across the cultivars. It's not exactly correlated year to year, but some of the cultivars had pretty low recovery, and some of them on the right had higher recovery. The pathogen was also moving across the vine. We inoculated all vines on the western side and then detected across the vine. And it was moving extremely slowly at first. So in the second year, in 2022, we could only detect in a few vines on the west center, east center, and east side. And similarly, in symptoms, so what you see on the bottom is the percentage of symptomatic vines. In 2021, the first year, there were only symptomatic vines on the west side. In the second year, 2022, most of the symptoms were concentrated on the west side, with a few on the west center, east center, and east. And then in 2023, you can see that the symptoms have finally evened out across the vine, indicating that the pathogen has been able to reach that eastern cordon and has spread to the east center and east side. We also saw some unique symptoms in these Mediterranean cultivars. Specifically, you see this dried out shoot in Tinta amarella, and then also in the inoculated shoot, these are all from the first year. You see a lot of leaf loss, and you see these shriveled clusters, which is a known symptom. And then by 2023, I talked about how the symptoms had shifted. You can see the western side of the vine, which is where we had inoculated, was not really pushing shoots anymore. And then the symptoms, so shriveled clusters and scorch, which were the predominant symptoms in September 2023, were actually more concentrated in the center of the vine, so west center and east center, than on the west side, which was an interesting observation. We were also able to measure stunting by actually taking a ruler and measuring the height of the vines early on. And what we see in 4 of the cultivars is that vines that were infected that year, so they have a 1 on the x-axis, had shorter average heights than vines that were uninfected, ones that have a 0 on the x-axis. And so early on in the season, you can actually measure delayed growth in some of these vines.
To build on the field trial, we inoculated plants from 9 of the 13 cultivars in the greenhouse in spring 2023. And here's just a comparison of the inoculation success rate between the field trial and the greenhouse. We wanted to verify the field trial's inoculation rates. And also, our goal is to build on this field trial in future work, and ensuring that the data from the field trial is similar to what we do in a controlled setting was really important. Next. After the summer, we transported the vines from the UC Berkeley greenhouses to Hopland. We keep them in screen houses over the winter, and so the goal here is to compare the overwinter survival rates across the cultivars to the data from the field trial as well. This method of greenhouse to screen house transportation of vines has been used in the past, and we plan to use it again in the future. So this is paving the way for future work looking into the recovery mechanism in this overwinter curing process.
Great. Thank you for listening, and we hope you learned and enjoyed listening to our research.